Greetings, students, and welcome to Chapter 8, the Cellular Basis of Reproduction and Inheritance. In this particular chapter, we're going to look at four big ideas. We're going to look at cell division and reproduction. We're going to look at the eukaryotic cell in terms of mitosis. Then we're going to cover meiosis, and then we're going to close out looking at alterations of chromosome number and structure. That'll be the last part of our chapter. So we'll start first with cell division and reproduction. Let's first of all discuss why cells reproduce. And this was something we talked about earlier in the semester. Cells reproduce for three main reasons. First, to allow for an organism to grow. I mean, think about when you were born. I know you don't remember, but think about how small you were and how big you are now. That's because you've added a lot of cells since then, and those cells have allowed you to continue to grow. To replace damaged cells and to make more organisms. Another reason why cells have to reproduce or divide is because, remember, there's limits to how big a cell can get. And we talked about this in the plasma membrane chapter. Remember, volume increases much faster than surface area. So if the cell were to continue to grow from the inside out, the cell membrane would not be able to keep up. Like a balloon that's continually being inflated, it would pop. Also, if cells get too big, it would be very difficult for the nucleus to manage everything that's going on. And it would also be very difficult for the cell to be able to move the materials in and out, especially as it tries to traverse that big distance all the way from the membrane into the center part of the cell, or from the center part of the cell out. So the cell solves this problem by dividing. Now, cell division is at the heart of the reproduction of cells and organisms because cells originate only from pre-existing cells. Remember that from when we talked about the cell theory? Some organisms reproduce asexually, which is one parent reproduction, and then other organisms reproduce sexually with two parents. Now, asexual reproduction is beneficial because it takes less time, you don't have to find a mate, that kind of stuff, but it's not as beneficial because you don't get the kind of variation that you do in sexual reproduction. You're getting a genetic clone, and in that case, the copy is only as good as the original, right? If an organism reproduces sexually, it might take a longer amount of time. I mean, you have to find a mate and stuff. And genetic information has to be exchanged and all that. But you do get a lot of variety in the offspring. And so things that might be wrong, there's more mechanisms in place to actually correct them in the offspring. Now, let's start first by talking about a simple type of reproduction that occurs in prokaryotes, which, of course, remember, are organisms that lack a nucleus. Prokaryotes, for the most part, reproduce asexually by a process called binary fission, which means dividing in half. And what will happen during this process is the cell will take that genetic information, which, remember, it's not in a nucleus. It's like a big circular piece of DNA, sometimes referred to as a plasmid. And what will happen is that DNA will be copied. The plasma membrane will pinch inward and tear, and then the new cell wall will be made, which will create two new daughter cells. So it looks something like this. Here's the genetic information. The genetic information is copied. The cell membrane starts to pinch inward, and then the cell wall will form on each one so that it divides and creates two daughter cells. This is a form of asexual reproduction because the genetic information is the same in the parent and the daughters. And if you look at the genetic information, it pretty much looks the same too, doesn't it? Here's an actual image of a prokaryotic cell undergoing binary fission. But for our chapter, we're going to concentrate primarily on those much more sophisticated and complex cells, the eukaryotic cells. So that's what we're going to be looking at for the bulk of the chapter here. Eukaryotic cells, of course, have more genes than prokaryotic cells. They have multiple chromosomes. So their process is a little bit more complicated. It starts first with the nucleus, of course. That's where the genetic information is located. That genetic information is stored, of course, in DNA. When that genetic information is in the nucleus and the cell is not under the process of dividing or preparing to divide, we call it chromatin. And these are very, very small pieces of genetic material. They're thin and fibrous, almost like swirled around like spaghetti. But when the cell gets the message that it's time to divide, that genetic material will actually condense together. And that's when you'll begin to see what we know as a chromosome. And what happens is when the chromosome forms and duplicates its genetic information, you end up with what are called sister chromatids. And sister chromatids are two pieces of identical DNA that are connected together. And what cell division really is about when the cell divides in two is about separating those sister chromatids so that each daughter cell ends up with the exact same genetic information. So here is the chromosome. After duplication takes place during interphase, you end up with two pieces of DNA, whereas you can see there was only one here, now there's two. But these are attached together. So here's one sister chromatid, here's the other, and they're attached together at this central point here called the centromere. Okay. After cell division takes place, those two sister chromatids will separate, as you can see here and here, and the DNA will move into two cells, and those two cells will become their own independent cells with their own nuclei. And here's another image of it here, zoomed in a little bit. And here's the chromosome zoomed in. You can see the centromere very clearly, and also the two sister chromatids. Now, what we're going to be looking at from this point on is how that whole process takes place. The whole process together is known collectively as the cell cycle. And the cell cycle is the orderly sequence of events that runs from the time a cell is first formed after it divides from its parent cell until it divides into its own two daughter cells. That whole cycle is the cell cycle, not just the part where the nuclei divides and all that. There's a whole cycle that runs during the life cycle of the cell. And it looks something like this. Okay, so if you imagine this pie chart here, you have all this time here in the light brown or tan color. That's called interphase. And interphase is really the bulk of the life cycle of a cell. And you can see it's like 80 to 90% of the cell life cycle. And then you have a small part here, like 20, 10%. That's actually the part of the time when the cell is actually dividing and undergoing the process of mitosis or the mitotic phase. Here's a better diagram here, and you can see the distinct stages here. You can see G1 in interphase, also known as gap 1. You can see the synthesis phase, and then you can see gap 2, which I'm going to talk about all these things in a second. And then lastly, the M phase, which contains all the stages of mitosis, all four of them, and then ends up with cytokinesis right here, which is the division of the cytoplasm. So now let's look at these steps a little more closely here. Okay. First of all, if we were to talk about this process of mitosis, which is actually the process of cellular division, it could be summed up very simply as this. Mitosis is where the duplicated chromosomes are separated into the two daughter nuclei. Okay? And it'll do that as the chromosomes move across what's called the mitotic spindle, which I'll show you in a second, which are made up of microtubules that allow the chromosomes to move across, back and forth across the cell and into the new daughter cells. And mitosis is really about the division of the nucleus and the separation of those sister chromatids into what ultimately will become two new nuclei. And this is what the cell cycle looks like in a nutshell. Starts first with interphase, which is that in-between phase when the cell is not dividing. Interphase has three parts: G1 or gap 1, which is the primary growth phase. That's the phase that is immediately after the cells have divided and gone through cytokinesis. This is where the cell is growing, adding additional organelles, all that kind of stuff. And then eventually in G1, the cell will get a signal that tells it's time to divide. And then the cell will move into the S phase, which is synthesis. And that's where the genetic information is replicated or duplicated so that the cell can divide. And then after that, we move into G2 or gap 2. In this stage here, this stage is where the cell is getting ready for division, producing the types of enzymes and proteins it needs to have to undergo mitosis. Then we move into mitosis, which is literally the division of the nucleus, and then finish up with cytokinesis, which is the division of the cytoplasm. So now let's look into the stages of mitosis itself. So we've gone through interphase, the DNA has been duplicated, all that stuff, and now we're going to be moving into actually the first part of mitosis. And you can remember mitosis stage by just thinking of this PMAT, which stands for prophase, metaphase, anaphase, and telophase. So the first stage of mitosis is prophase. In prophase, the nucleolus will disappear, chromosomes will condense, which means they'll coil up and kind of take on that distinct shape we think of when we think of a chromosome. Centrioles will move to opposite poles. These are the poles here, and those are the centrioles. Spindle fibers will begin to form. These are the microtubules I mentioned a few minutes ago, which will facilitate the movement of the chromosomes. And then the nuclear membrane will begin to break down. And it has to break down when you think about it, because it's got to release the genetic information on the inside. And so this is what prophase looks like. Then we move into metaphase. In metaphase, the chromosomes will line up along the equator here, or what's called the equatorial plate, or sometimes the metaphase plate. At this point here, the centromeres are attached to these spindle fibers right here. You can see each centromere is attached to a spindle fiber. Okay. And the spindle fibers are kind of moving around a little bit. They're vibrating and stuffing, and there's kind of like a tug of war going on as it tries to pull apart the sister chromatids. And once it's successful, the sister chromatids will actually separate apart. And so this is one set of chromatids that you can see here, and this is the other set of chromatids. They were once attached at the centromere, now they've been separated apart, and each one is going to move in the direction of the pole during anaphase. And you can see it in this very clearly in this plant cell picture right here. The DNA is now separating into two, what will ultimately become the two new daughter cells. Each cell will have the same amount of genetic information, and it will be identical in both. So that's anaphase. Then anaphase moves into telophase. In telophase, the chromosomes will start to uncoil and begin to take their more traditional look as they begin to get ready to become chromatin again. New nuclear membranes will reform, but now two of them will form, because each one of these eventual daughter cells will have their own nucleus. And in this particular point here, and probably towards the end of anaphase, cytokinesis will begin. And cytokinesis is when the cell starts to pinch and start to get ready to tear, which I'll talk about more in a couple minutes. In animal cells, the cell will kind of twist, pinch in, and tear. Whereas in plant cells, they will grow a cell plate, which will ultimately become the cell wall. And I'll show you more of that in a moment. Okay. I do want to highlight these two slides here, 8.51 and 8.56. I kind of removed some of the other stuff in between. These are two really great slides that highlight really nicely each stage of the process of mitosis, so you can really get familiar with what they look like. So if there was, say, a test question, you might be asked to identify the stage. This would be a really good thing to study. Now, once the cells are done with the process of separating out the genetic information, now the cells actually have to separate from each other, and that's cytokinesis. Okay? And this is ultimately when the cell will finally completely divide into two. In animal cells, cytokinesis will occur in such a way that the cell will constrict or pinch together, forming what's called a cleavage furrow, which I'll show you in a second. And in plant cells, they'll produce kind of a membranous structure called a cell plate, which will ultimately become the cell wall, which will divide them in two. So in an animal cell, and that's what this is, an animal cell, you can see the cleavage furrow right here. This is the cleavage furrow, that kind of pinching structure where it's pinched in and it's kind of contracting in. This is where there's a ring of microfilaments that are kind of squeezing the cell together, kind of pinching it together and helping facilitate it tear apart into the two daughter cells. And here's a close-up look from electron microscope. Here's a couple more animations here, drawings. And in a plant cell, you can see here, and on the left-hand side is an actual light microscope image, but you can see on the right here, the formation of the vesicles, which are carrying the components to make a cell wall. And so what will first form is this kind of cell plate here, which eventually will grow out and ultimately form into the new cell wall, which will then divide the two cells completely into two separate daughter cells. Okay. And this is what the process looks like. I'll show you real quick in a short clip, and that's how we'll close out this part of the lesson.
In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.